Hello everyone, this is the second part of attenuated vaccine, and in this part we learn about the approaches or process for the production of live attenuated vaccines. And as I said in the last video that there are two type of processes, classical process and modern genetic engineering process. So first we learn about the classical process. There are different classical process or approaches by which we can produce a live attenuated vaccine. Okay. So now I'll talk about those. So first, use of naturally attenuated vaccine. Okay. So and this approach is based on variation in virulence among human viruses belonging to the same serotype. For example, type 2 poliovirus strain on the live oral poliovirus vaccine is a naturally attenuated vaccine. I mean the vaccine which is used, the poliovirus vaccine is actually a naturally attenuated live vaccine. Okay. However, I mean, in this case, I mean, if we want to get a naturally attenuated virus, we really have to be very lucky. Okay. Then another approach, the use of related virus from another animal or Jennerian approach. It is called Jennerian approach because this approach was introduced by Edward Jenner, and we know he was first. He was the first who invented the pox virus, right? And what was the approach? I mean, he wanted to, uh, I mean, protect human against smallpox. That means he wanted to induce immunity against the smallpox in humans, but as a vaccine he didn't use a smallpox. Rather, he used a related virus. He uses the cowpox virus, which don't cause disease in the human but can induce immunity in human, which protect the human against a smallpox infection. Okay. And another approach, passage of virulent virus in an unnatural host or host cell. Okay. For example, the live attenuated measles virus currently in use was derived following serial passage through the human kidney, human amnion, sheep kidney, and chick embryo cell. So, what we have to do if in this approach? So, I mean, we have to culture everything, right? If it's a bacteria, we need to culture if we want to keep them alive. It's also true for virus. If we want to keep them alive, we need to culture them. And for as like for bacteria, we can culture them on nutrient media. But virus, we know those are obligate intercellular parasites. So for virus, we need to culture them on cell, right? So in this approach, what we do, we culture the virulent virus in their unnatural cell. Unnatural cells means, for example, the measles virus. Measles infect the respiratory tract, the lower respiratory system. I mean, measles infect the epithelial cell of the bronchi. Okay, and the epithelial cell of the lower respiratory tract. So, to make it attenuated, virulent measles virus was cultured and passaged in human kidney cell, for example. So human kidney is not a natural infection tissue of the measles virus. Measles virus don't infect human kidney cell. Okay, so by doing so, culturing and passaging the virulent virus in its unnatural host cell may make it attenuated. Okay, and actually the measles virus, okay, which is currently in use, was attenuated in this way. Then another approach, modification of conditions. Okay, so in this approach, attenuation can be achieved by growing a pathogenic bacterium or virus for a prolonged period under abnormal culture conditions. I mean, we have to culture the bacteria or virus in not in their natural environmental conditions, rather in abnormal uh, culture conditions. Okay, and these procedures cause some mutation in the bacteria or virus. Okay, and if we are lucky enough, we will get a mutant which will survive in that abnormal condition, but will lose its virulent factors. Okay, and if we can get that mutant, we can use that mutant virus or bacteria as a vaccine. So, for example, an attenuated strain of Mycobacterium bovis called Bacillus calmette-guerin, BCG, the BCG vaccine. Okay, this famous BCG vaccine was developed by growing Mycobacterium bovis on a medium containing increasing concentration of bile. It is an abnormal condition for the M. bovis. Okay, however, it took 13 years. Okay, 13 years. Imagine how long it can take. So after long 13 years, this strain, the strain of M. bovis, had adapted to growth in a strong bile and become attenuated. Okay, and that is used as a vaccine for tuberculosis. Okay, it took long 13 years. Wow. Then another approach, genetic reassortment. Okay, so what we do in this approach, that is genetic reassortment. So what we do in genetic reassortment approach or process, mixed infections with pathogenic and non-pathogenic viruses with segmented genomes have been found to give rise reassortments with desired attenuated properties. So what we do in this approach, we have to infect the host with pathogenic, we have to infect the host at the same time with pathogenic and non-pathogenic virus. So I mean, after the infection, we can get some strain of that virus which is attenuated. For example, this approach, that is my genetic reassortment approach, is used to develop influenza vaccine. Okay, so how? Reassortments between temperature sensitive mutants of influenza, this is avirulent, okay, the TS mutants of influenza, and currently circulating strains are selected. Okay, and if you want to develop, in this approach, if you want to develop a vaccine against the currently circulating strains of influenza virus, what you have to do, you have to infect the host at the same time with currently circulating strain and with temperature sensitive mutants of influenza, and you have to select the hybrid. Okay, so which hybrid? Those who maintain the TS phenotype, temperature sensitive phenotype, and at the same time have the H and Na proteins of the currently circulating strain. And H and Na is these antigens are expressed or found in the surface of the influenza virus and can induce immunity. Okay, can induce our immune system. Okay, so by this way we can get a attenuated influenza virus, okay, which cannot cause the disease, but yet as it has the H and Na in their surface, they can induce immunity. Okay, then another approach, administration of pathogenic or partially attenuation virus by an unnatural route. Okay, so in this approach, what you do, suppose a virus, it infect the lung, respiratory system. So we would administer this pathogen not in the lung but in the stomach. Okay, in another route. Okay, in an unnatural route. Okay, so according to this approach, attenuation is achieved by limiting infection to an area of the body in which disease does not develop. Okay, for example, live adenovirus vaccine administered orally in an enteric coated tablet establishes an asymptomatic immunization infection that does not spread to the respiratory tract, which is the region of nasal infection. In this approach, you want to vaccinate a person with adenovirus. You want to induce immunity against immunity against adenovirus in a person. So what we can do, you can administer enteric coated adenovirus, okay, into that person's stomach. Okay, just how you can do that, if you just feed, just orally administer your vaccine. Okay, and you have to actually coat your adenovirus, otherwise it will get digested in the stomach. Okay, anyway, so once you orally administer the adenovirus vaccine, it will then cause a asymptomatic infection in the stomach. Okay, so it will, and due to this asymptomatic infection, it will induce immunity against the adenovirus into the host's body, but this infection can never reach to the respiratory tract. That means it will not spread in the respiratory tract. Okay. So by this way we can avoid the infection, at the same time we can induce the immune system. Okay. So this was the these were the classical process for live attenuated vaccine production. In the next video lecture, I'll talk about the genetic engineering approach for production of live attenuated vaccine. Okay, thank you.